First, let me introduce myself. My name is Stefano Lonardi. I'm a professor at UC Riverside, which is not too far from here, on your way to Palm Springs. And I've been working on computational biology in different forms since the late 90s, like we need. Assembly has been sort of one of my favorite problems to work on and off over the years. And today, what I would like to talk about is looking at this problem from a more practical point of view. I know if someone gives you some data, how do you go about getting a good assembly? Why do we still care about assembly? As we heard from Ryan's talk, there is a lot of species on the planet that for which we haven't got the genome. Probably, the number of genomes in at different level of completions is maybe probably 20,000, 30,000, depending how you want to count it. Genome assembly is still very hard, right? You can get a good raft very quickly, but finishing it takes quite a bit of effort and time and sometimes money. And that's what I would like to talk about today. How do you finish in a genome in a reasonable amount of time? So a quick reminder: this talk is a little bit tutorial/research. So we have 3 generations of sequencing instruments on the market. The first generation, which is Sanger, very few people are using it unless these sequencing instruments are still on campuses. The read length is about 1,000 base pairs. Then the second generation, which up to 300, around that number, which is typically Lumina. And then we have the third generation, which is essentially at this point is PacBio and Oxford Nanopore. Read lengths typically 50K. Then depending on Oxford Nanopores are longer. It can go up to sometimes 1 million, 2 million base pairs, but they tend to be very noisy. Still, they don't compare. They're still way too short, right, to read the chromosome from the beginning to the end. And so we have this idea of taking the genome, shredding it into pieces, and then sequence each piece individually. And then the problem of genome assembly is to put it together. It's a big puzzle. So these fragments are called reads, and they need to be assembled by detecting overlaps and using other tricks that we can leverage to get a good assembly. So in inputs, typically you're given either long read or short read or both, and these are some of the technology that are available on the market. And ideally, you would like to have some sort of a map. In the form of physical in the old days. These days would be a genetic map or an optical map or maybe a HiFi map, right? So you generate, for example, IC reads from proximity ligation experiments, and this gives you this long-range information between the mates. The output of this problem is we want the most contiguous and error-free assembly possible. So one of the arguments that I would like to make today is that these two objectives are playing against each other, because if you don't do anything to the reads and you don't assemble them, obviously you're not going to make any mistakes. As soon as you start assembling them, you increase your chances of making mistakes. So of course, you get longer and longer pieces of the genome, but you increase this probability of misassemblies. So optimizing both of them is a trade-off. So it depends on how comfortable you are in terms of creating errors in your assembly. At this point, there is no assembler that gives you the perfect assembly every time from beginning to the end. All right, so today I would like to focus on HiFi reads, and I'll talk a little bit about optical maps. Mostly because of our experience with these types of mappings. All right, so overview again. You have reads. You try to find overlaps. You create these longer pieces of the chromosomes called contigs, and then we try. Once you have these contigs, these long arrows, you want to figure out what the distance is between them, and the relative orientation, right? Because sometimes you sequence the 5 primes, the 4 strand, sometimes you sequence the reverse strand, so they can be in reverse relative orientation. So these relations between the contigs can be obtained by using mate pairs, pair and reads, high-C reads, optical maps. So what you end up with is what's called a scaffold. So you take these contigs, you place them, you figure out the distance between them, the relative orientation, and now you have a sequence, a gap, a sequence, a gap, and a sequence, and that's called a scaffold. These gaps are unassembled typically because these regions are repetitive, and we're going to talk more about why the repetitive regions create an issue for modern assemblers. The goal, again, is to maximize contiguity and minimize the errors. So in these slides, I try to sort of summarize what types of actions you can do pre-assembly and post-assembly. Again, in input, you're given different technologies, might be given also some maps. So in terms of pre-assembly, sometimes we do error correction. Sometimes we trim the reads, if the Illumina reads. Sometimes we remove maybe the end, the low-quality portions of the reads. If you have a lot of data, a lot of input reads, which is rare, but it happens if your genome is small and you're sequencing a lot, you might need to do read selection, right? So if your coverage is really, really high, you might need to figure out which of the reads you want to use for the assembly, and that's not an easy problem to solve. You need to choose an assembler, and I'm going to talk about that in a minute because there is a lot of choices. You will get these contigs, and then from the contigs, you might use these maps to create scaffolds, which is your final assembly. However, there are other things you can do post-assembly that typically people do, including trying to do error correction, correcting the misjoins, detecting and correcting scaffolds which were incorrectly assembled, maybe using an optical map, and I will show you some example later. But sometimes we could try to stitch them. If you have multiple assemblies, you might want to polish the assembly using maybe luminaries, which high quality. And there are some attempts also to do gap filling, right? So those gaps that I show you with all the ends, you might want to see if you can actually fill them in. Okay. These are all separate tools. So there is a lot of choices here, okay? Lots of choices, and that's why this is tricky, right? So there are pre-assembly choices that you want to make. You need to choose an assembler, and then you have all these tools in post-assembly that you need to choose from and decide whether to use them or not. So the challenges come from two aspects. One is technology dependent, which is related to the sequencing instruments. Sequencing instruments are great. They've done immense progress in the last 20 years, 25 years, 30 years in terms of throughput, length, and sequencing error, but they're not perfect. We have sometimes uneven sequence coverage, meaning there are some regions of the genomes that do not have enough reads or very few reads. There are some sequencing errors, in particular very, very long reads. And sometimes these errors are non-uniform. They tend to cluster in certain positions or for certain biases. From the genome point of view, the issues are mostly due to the fact that we have genomes that tend to be repetitive, right? Unfortunately, that's a big issue for genome assemblers. So in this picture here, this is an old picture from BioNano, shows the amount of repetitive regions and the copy numbers. So these are tandem repeats or segmental duplications in these 7 species. And you can see this is the size, the repeat size unit. And on the y-axis, you have the number of copies of that repeat. So you could have thousands of copies repeated. So a unit, a monomer of 10 kb repeated 1,000 times, for example. And those, you can imagine how hard that to assemble, because if you get a read in that region, they're going to overlap with each other crazily, right? They're going to be the same string. The other issue is in some genomes, and this is when we sort of move away from the human genome, which is important, but there's only one species over 10 million species on the planet. There are genomes which are tetraploid, dextraploid, so there are many copies of the same chromosomes, and there are small variations between them, and reconstructing them is really, really hard, reconstructing every copy of the chromosome. How are we doing? So this is a nice paper that came out in Nature Plants just a few months ago, and it shows that they did an immense amount of work. They collected data for about 2,300 assemblies, plant assemblies. They plotted on so the x-axis is the year of release of that genome, and on the y-axis, the size, the M50 size, which is a measure of contiguity. Okay? So the M50 means that 50% of the assembly is contained on that value of the contig or larger. So if you sum up all those contig sizes, you get 50% of the assembly. So the larger, the better. And this is a log scale. And what they did is they have lots and lots of figures, and this was probably the most interesting one. You can see the different technology over time. So these blue ones on here is mostly Illumina, and then these green ones here is mostly regular PacBio or Oxford Nanopore. And then recently, these light blue ones, this is HiFi. And as you can see, recently, HiFi dominates assemblies. Okay? Everybody is choosing HiFi, right, to make plant assemblies. You can also see a trend in getting the M50 higher and higher, which is good. But we are not getting to the point in which the M50 is the size of the chromosome. Okay. Another interesting aspect that they pointed out, again, they collect they put together a database for this, is what was the most used assembler and the most used polisher and the most used scaffolder. So HiFi as was the most predominantly used for contig assembly. And we had the talk by you a couple of days ago on the tool that he put together. And then NextPolish was the polisher, and 3D was the scaffolding tool that is based on IC. These are my very small list of genomes that we worked on. Oh, there's a fly here. Come on. All right. What's going on? All right, so the last one is this Babejo Rancani that we sequenced about 2-3 years ago using PacBio HiFi, IC, and optical maps. Just to give you an idea about the pipeline, we used a bunch of tools, again, for quality control, the assembly. At that time, I don't know why we didn't look Hifiasm, but certainly we should have. We're using polypolish for polishing, and then kinds of things to do the assembly quality control. The roadmap of my talk would like to talk a little bit about the assembler to use, and then talk about assembly reconciliations, and then detecting, correcting misjoins, and then dealing with repetitive regions a little bit at the end. So there are two papers that I would like to mention about which assembler to use. There is this paper in Nature in 2022 by Jarvis et al. That looked at different pipelines to reconstruct the human pangenome. So they look at pipelines using Canoe, iCanoe, cross-stitch, DeepIAS, and so on and so forth, Peregrine, Shasta. And one of the sentences in the paper, it says that when they assemble the human genome and they compare these human genome against HiCM bionano maps, they saw all kinds of problems, right? Misjoins, inversions, duplications. And one of the statements is that there is no single pipeline that works the best, that works on everything. Okay? By the way, recently I looked at 80002, which is another important assembly for the human genome. That was done with Verco using both HiFi and Oxford Nanopore. And version 0.7 that was published in 2022 still had very big gaps, right? 9 gaps related to the rDNA regions. And then there was tens of thousands of patches to get to version 1.0. And that took almost a year to get there. So again, assembly is hard. Okay? It takes time. Takes effort. The other paper I would like to mention is these paper that we published just a few months ago in which we evaluated several assemblers, just assemblers. This is different from Jarvis. Jarvis was about the pipeline, and here we're just measuring assemblers. So we used 3 plants: rice, homozygous type diploid, so essentially an haploid, potato, which is a diploid, and then we used apple, which is a tetraploid, and then we used humans, which is heterozygous diploid. We did measure these. We did tested Hifiasm, like canoe, fly, micat, miniasm, and so on and so forth on all these data sets, including 25 synthetic HiFi reads. And we measure all kinds of things, including not just the contiguity, but also time, memory, quality of the assembly. There is a lot of pictures in the paper. I don't have time to go through all of them. So this is one of the main pictures, main figures of the paper, in which we look at rice, potato, and wax apple. And on the left side, we are trying to plot the size of the contigs that you want. So you want contigs which are really long, so dark blue, right? And then you want the number of contigs, which is represented on the right side, to be as small as possible. This bar here, this dotted line is the actual size of the genome. So for example, if you look at next de novo actually generates good assemblies for rice. You can see the number of contigs is really small, which is great. However, when you go to potato and wax apple, there is a lot of the genome which is missing. Okay, the genome is 1.6 gigabases, and you only assemble maybe about one. Okay. On the other hand, Hifiasm is consistently good across these 3 data sets. We have long contigs, very few contigs, but very long, close to the genome size. So you can look at these types of papers, benchmarking papers, to decide what assembler would be the best for you. We also look at quality, obviously. So these are the quality values for these assemblies, and many of them actually achieve a very good qualities. Some of them are poorly generating assembly. On humans, we also look at human genome or human data because the reviewer wanted to. We tested the same set of assemblers. We included LJA and Rust. And again, you can look at these figures and decide which one you think would be the best assembler for you. We also look, of course, at completeness using Basco and so on and so forth. First conclusions about these types of analysis. Benchmarking helps, but they have limitations. They only benchmark those assemblers for those specific genomes. And sometimes it's hard to generalize, right, to make conclusions that apply to your own gene, the genome that you want to sequence. And there is no assembler that works every time perfectly. There are some good assemblers that generate good assemblies consistently, but there are pros and cons for different assemblers. So one of the ideas that we had at some point that was actually proposed a long time ago was why not generate many assemblers? So since we don't know which one to pick of all these assemblers, why don't we generate multiple assemblies with multiple assemblers? So we'll run Hifiasm, Canoe, and Verco, and all the other ones. We get n assemblies, and we try to put them together, trying to merge them. Okay. So there was a lot of work on this maybe 5 to 10 years ago to try to merge assemblies, maybe using the reads, maybe not. It turns out that it's really hard to do, okay, because you don't know which assembler to trust, essentially. So we published a paper in Genome Biology now a few years ago in which we tested these tools that are supposedly taking all these assemblies and merge them and get a better assembly for you. They really don't work. Unfortunately, they don't work. What can be done, though, is use the help of an optical map and use the optical map to merge assemblies. So we showed this a few years ago. So let me tell you a little bit the optical maps. In optical maps, what you do is you take your long piece of DNA, usually megabases long, and then you have nicking enzymes that recognize a specific extraction enzyme, in this case CTTAG, and then include the fluorophore in the DNA. Okay? These fluorophores then are imaged through a nanochannel, and then you can essentially get the distance between these fluorophore positions. That's all you get. You get the distance between these positions, and this fly really likes me. And then you can use this map to scaffold, for example, or correct your assembly. So what we did now 6 years ago, was to create multiple assemblies using different assemblers. At that time, we were not using these ones, but other ones. And then the first step was to map these assemblies against the optical map. So in this case, the optical map is in green. These black lines indicate the position of the restriction enzymes. So as long as you have a very nice alignment, it means that your assembly is consistent with the optical map. And then as you can see, sometimes you will see these positions where there is a break, right? So at this position, your contig does not agree with the optical map anymore, and therefore you can decide, you'll have to decide whether to cut this or not. Right? This could be a misjoin, okay? And the same is true for this second scaffold, contig. We have a tool that allows you to do that. You input the optical map, you input a scaffold, and then the tool will tell you there is a misjoin in this position. But the fun part is when you have multiple assemblies. Maybe you have 5, 6, 10 assemblies produced by different assemblers. What you can do is you can align them to the optical map, and then sort of pick and choose the contig that you prefer, the contig that seems the best for that location in the genome. So you don't have to pick one of these assemblers and do the whole assembly with the same assembler, but you can locally choose one assembler, right, or another. So what we do is we find the minimum tiling path of all these assemblies, again, guided by the mapping against the optical map, and then we stitch them. Right? We take these two contigs here that are sort of highlighted in red, and we stitch them. We know that they're overlapping the same optical molecule, so they must be overlapping, and they are, and so we can stitch them and generate a much longer contig. And it actually works very well. This is actually very relatively simple, but works very well. Of course, you need an optical map. If you don't, then this problem is really hard to do. So this is called novo and stitch, and you can check it out if you want. There is a lot of algorithmic details that, of course, I skipped. The last thing I want to talk about is repetitive region. So as it was mentioned many times, even yesterday in Pavel's talk, when you have tandem repeats, when you have the same monomer repeated over and over and over again many times, in this case t times, what you end up with when you do an assembly is you get one copy, because the reads coming from these regions tend to overcollapse that region. They all align with each other, and then you end up only one copy, maybe two. Okay, so typical assemblers, good recent assemblers do better than this, but tend to overcollapse these repeats. And so what we want is we would like to get these T copies out of the genomes. So we're assuming, as Pavel was mentioning yesterday, that each copy of these monomers, these alphas, has small variations between them. If you don't, there is nothing you can do. Right? If there is no variation between these copies, if they're identical to each other, either you have a read that covers this region entirely, otherwise you will never be able to solve this problem. You need very long reads. So the idea is to use these small variations between the copies to try to resolve these regions. So what we do is we have a draft genome, right? So this is an assembler that is entirely focused on resolving repeats. It's not a general assembler. So we take a draft genome like this one, we map the reads, we map HiFi reads in this case, against the draft genome and realize that, okay, so the coverage of this region is, say, 10 times bigger than the average coverage, and therefore it's likely that we have 10 copies of alpha here, right, which are over-collapsed. So in the first step, we identify these repetitive regions, and typically these are centromeres, but not just centromeres. Sometimes there is also highly repetitive regions which are non-centromeric. Here is an example. This is an Arabidopsis. So if you map the HiFi reads against the Arabidopsis genome, you get this big spike in chromosome 5 in these locations. And these coverage spikes actually is a well-known region which contains 45 copies of a unit which is 4.5 kb. Okay. So lots of copies. So, and the next step is to use uniquemers. And this idea of using single copy k-mers sort of has been floating around for a while now. The first time we find the reference to this idea is in this paper in Science from Evan Eckler lab. So what are these uniquemers or single copy k-mers? These are k-mers that appear only once in the genome. Okay. Pavel yesterday was talking about substrings that appears only once in the genome to, again, being able to distinguish these copies of the repetitive region. We do the same thing here, but because we're dealing with the reads, what we need to do is we need to look in the reads and find a k-mer that appears on average. So the number of times it appears has to be equal approximately to the average coverage. So if your data is 40x, so if you're covering every base pairs of your genome 40 times on average, then a uniquemer will be a k-mer that appears about 40 times plus or minus something, right? Because the expectation is that you have uniform coverage. So this idea was also used in Centrofly. And then also in the assembly of chromosome 8. So Glynis Logsdon is actually a collaborator in this project, and she used it to finish the centromere of chromosome 8 that was published in Nature in 2021. Okay, so in step 2, we take these reads that we selected by determining that there is a spike in coverage, right? So we take these red reads, we look for uniquemers, which are k-mers in this range, mu minus t times sigma, where sigma is the standard deviation and mu is the average coverage. And then what we do is, this is just, so we get these uniquemers, and then what we do is we barcode the reads. We take the reads, we ignore everything except the position of these uniquemers, and then we use these uniquemers to determine overlaps. Okay. So we barcode the reads. Of course, we have to look at the reverse complement as well. And so we create clusters. So the idea is we hope that each cluster will represent one monomer, one copy of that repetitive region. And so these two reads are together because they share the same uniquemers or the same barcode, and these two are also together. Sometimes you'll have only a few reads in it. Once we have these groups of reads, then we can assemble them individually. So we take a regular assembler, and we create context for each of these clusters. Then we use Minimap to compute overlaps. We build an overlap graph or a string graph, and then we generate an assembly out of these overlap graph. We'll build the consensus. We tried this on different genomes, not just human, but I would like to show you the human genome result. So what we did was to assume we went back in time. So we took the assembly GRCH38, which was the pre-T2T assembly for the human genome, which, as we know, was missing about 8% of the genome, mostly repetitive regions. We took HiFi reads, modern HiFi reads, and we asked ourselves, can we take H38, can we make it better? Can we resolve some of these repetitive regions? So we call these assembly RA38 in the next few figures. And then we evaluated RA38, this assembly using Quast, NUCFR, Crack, and Siri. And then we used the T2T assembly as the ground truth. So the idea, again, was if we were back 5-6 years ago when GRC H38 was the state of the art, could we make it better? Again, you need HiFi reads, right? Because these unique mirrors, you can use this idea of unique mirrors only if you have very, very high quality reads, right? And HiFi reads fill the bill. So first we try Crack. So Crack is a tool that you're given an input on your assembly and HiFi reads or Illumina reads or other types of reads, and it will give you scores for the quality of the assembly. So it will compute some concordance scores between the reads and the assembly that you have, so it's reference free. Okay, so in this case, we did not compare our assembly with T2T. But clearly, the coverage improved, obviously, because we look at we're trying to improve the chromosomes centromeres for chromosome 8 and X, and the non-centromeric regions of 19. But the quality is also, these quality scores are actually improved. When we did Siri, in Siri you take two assemblies and look at synteny between the two assemblies. So on the left we have the synteny between AG38 and our RAmbler assembler. And this is the RAmbler assembler versus the T2T. And as you can see, the agreement between the RAmbler assembler and T2T is really good in these centromeres. Okay, so these are the centromeric regions where you have these inversions. And then once you do the full chromosome synteny, these assemblies look really good. We also look at NUCMR. So we showed that the frequency, the coverage, right, after you repeat the repetitive regions actually decreases significantly, which means that we resolved most of the copies, maybe not all of the copies. And then we look at, also we look at Crack in terms of quality. So I think I'm done a little early. So how do you, if you have HiFi reads, how do you get the best assemblies? First thing, you need to accept the fact that assembly is a compromise, right? It's a compromise between quality and contiguity. Read benchmarking papers can help reading them because it will tell you, for a particular genome size, for a particular polyploidity, for a particular heterozygosity, for a particular, whatever is your genome that you have, what would be the best assembler. HiFi-ASM is really good based on what we tested. Assembly reconciliation, very tempting, right? The idea of, oh, let's not choose one assembler, let's get n assemblers and trying to merge assemblies. It doesn't work very well at the sequence level, but if you do have an optical map, you can do that. So you can, with optical maps, you can, which is additional money, obviously, but you can identify chimeric contigs, assembly reconciliations. You can scaffold, obviously, and you can also estimate sometimes how much of the assembly is missing. Right? There's one of the biggest problems when you do an assembly is that you get this file with your contigs and scaffolds, but you still don't know how much of the genome you're missing. And that's the sort of the hard reality at the end. There is a little bit of hope in terms of resolving repeats, including Centrofly, Phase Dancer, and our RAmbler. So I'll stop here. Of course, all this work was done by my graduate students who are highlighted up here, and then lots of collaborators on different genomes and different methods. The code is on our GitHub page for Novo and Stitch and for RAmbler, and these are the references, and these QR codes will take you there. Well, thank you for your attention. I'll take any question you might have.